So what is an ISI? Basically, an ISI is all, the place where you put all your immunogenicity data. So throughout the ECTD, you'll have different places where we'll be detailing your immunogenicity summaries, but the ISI is where you put everything, and they're required. They have 4 key parts, and you have your analysis of your risk factors, your assay strategy, clinical trial design, your results, and then your REMS and conclusions. And I haven't really seen this in practice as much as I would like, but the agencies have asked that this be a living document. So as you're developing the drug, you are building and updating your ISI as you go along, not just writing it at the end. So I'm going to briefly go through the organization of the ECTD and what's in there. So 2724 is basically a brief discovery or brief description of everything. So, a little bit about the assays, the relationships of the disease to your immunogenicity results, your therapeutic dosing schedule and how that can affect immunogenicity. Also, what's clinically relevant. 253 is specific to the interpretation of the results. So your covariate analysis, your tabular summaries of your results. And then 5314 is the details of your validation and assays that were used across the different clinical studies. The ISI itself is added to section 5353 with the ISE, the Integrated Summary of Efficacy and Integrated Summary of Safety, ISS. So what is in the risk assessment? So the first part of your ISI, you're going to talk about the drug class, the route of administration, any patient disease-related factors, what's the likelihood of developing an immune response, what are the consequences to the patient? Is it just that this drug doesn't work for them anymore, or is there a potential for further adverse events if you develop an immune response to an endogenous protein? And what's the reversibility of the response? Does the ADA persist for a long time, or does it go away? So the next part of the ISI is going to be your assay strategy and your clinical trial design. You want to give your rationale for your assay method choice. Do you have a assay method to look at each part of the therapeutic? So, as we have a lot of different complex biologics out there. Do you have, if you have PEG, do you have an assay against the PEG? If it's gene therapy, there's going to be a lot of different ADA assays that you have to have against the vector and the resultant protein. Your tiered strategy, did you have any change in cut points? Was there any differences between patient populations? And then what's your assay life cycle plans? And then you're going to go ahead and lay out your trial design, your sample justification regimen, justification for your sample regimen. How did it affect PK? How did it affect PD and safety? Your overall results. So you're going to look at your covariate analysis. And that's what lots of times I set up my TLFs. I'll have, for my PK parameters, ADA positive patients versus ADA negative patients. For AUC, Cmax, et cetera. How did ADA affect safety? Did you see more adverse events in ADA positive patients? And also for efficacy, if you have PK clearing ADA, you're going to see less efficacy in those patients. And then you want to stratify your results, your ADA titer, your NAB status, your NAB titer as well. And, for some of these things, say for like a molecule that has PEG, did you have pre-existing antibodies? What were your pre-dose positive samples? And then you want to, have the traceability of the drug lots. Was there any difference in the drug lots that were used throughout the development process? And then finally, the fourth part of the ISI is your conclusions and your REMS. How does the ADA impact the subject population? How are you going to manage risk? How are you going to detect immunogenicity going forward? Linking these adverse events to immunogenicity post-marketing is also required, and the FDA also wants to understand your life cycle management. Who's going to run the assay? How are you going to keep it going? What's the assay requalification schedule? And so that you have it available for post-marketing surveillance. So in summary, for ISI, have all the risks been identified and described, were the assays appropriate to monitor your immune responses, were the studies and sampling appropriately designed, what's the incidence and persistence of ADA, how did it correlate to your PK, PD, safety, and efficacy, how do you plan to monitor immunogenicity post-marketing, you want this to be a living document, and it's required.